Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start a topic, please like, share, and subscribe the channel. In the last session, we dealt with enzyme kinetics. In today's session, we will deal with factors influencing enzyme activity. Previously, we studied Michaelis Menten kinetics and double reciprocal plot. Today, we will study Dixon plot. Dixon plot. The velocity v is measured at several concentrations of inhibitor I when the substrate concentration is kept constant. It is used for determining inhibition constants. A plot of 1 by v versus I yields a straight line. The experiment is repeated at different concentrations of substrate. Next is cooperative binding. Some enzymes may not strictly follow the Michaelis-Menten kinetics. When the enzyme has many subunits and binding of one substrate to one unit enhances the affinity for binding other subunits, that is cooperative binding, a sigmoid-shaped saturation curve is obtained. In this graph, we can see that sigmoid-shaped substrate saturation kinetics of cooperative binding is seen. In such cases, the determination of Km value as shown in the previous graph will be invalid. Instead, the Hill equation originally described for explaining the oxygen binding to the hemoglobin is employed. Third factor is effect of concentration of products. In a reversible reaction, substrate bidirectionally equal to product when equilibrium is reached as per the law of mass action. The reaction rate is slowed down. So when the product concentration is increased, the reaction is slowed, stopped, or even reversed. In inborn error of metabolism, the enzyme of a metabolic pathway is blocked. For example, A undergoes B in the presence of E1, then B undergoes C in the presence of enzyme E2, and then C undergoes D in the presence of enzyme E3. If E3 enzyme is absent, C will accumulate, which in turn will inhibit E2. Consequently, in the course of time, the whole pathway is blocked. Factor 4 is effect of temperature. The velocity of enzyme reaction increases when the temperature of the medium is increased. It reaches a maximum and then falls. This is known as bell-shaped curve. The temperature at which maximum amount of substrate is converted to the product per unit time is called the optimum temperature. In this graph, we can see that temperature is plotted against velocity, where bell-shaped curve is obtained, and optimum range of temperature is seen in between. As the temperature is increased, more molecules get activation energy or molecules are at increased rate of motion, so their collision properties are increased, and so the reaction velocity is also increased. The temperature coefficient, that is Q10, is factor by which the rate of catalysis is increased by rise in 10 degrees centigrade. Generally, the rate of reaction of most enzymes will double by a rise in 10 degrees centigrade. But when the temperature is more than 50 degrees centigrade, heat denaturation occurs and consequently loss of tertiary protein structure occurs. So activity of the enzyme is decreased. Most human enzymes have the optimum temperature or around 37 degrees centigrade. Certain bacteria living in hot springs will have temperature move with optimum temperature nearing 100. Next factor is pH. Each enzyme has an optimum pH on both sides of which the velocity will be drastically reduced. The graph will show a bell-shaped curve. The pH decides a change in the amino acid residues at the active site. In this graph, we can see that pH is plotted against velocity, and bell-shaped curves of pepsin, amylase, and arginase are obtained. The net charge on the enzyme protein would influence the substrate binding and catalytic activity. So optimum pH may vary depending on the temperature, concentration of substrate, and presence of ions. Usually, the enzymes have the optimum pH between 6 to 8. Some important exceptions are there, like pepsin with the optimum pH around 1 to 2, then alkaline phosphatase where optimum pH is around 9 to 10, and acid phosphatase whose optimum pH is around 4 to 5. Next factor is enzyme activation. In the presence of certain inorganic ions, some enzymes show higher activity. These chloride ions activate salivary amylase, and calcium activate the lipases. Another type of activation is the conversion of an important proenzyme or zymogen to the active enzyme by splitting a single peptide bond and removal of a small polypeptide from trypsinogen. The active trypsin is formed. This results in unmasking of the active center. Similarly, trypsin activates chymotrypsinogen to form active chymotrypsin and two peptides, A and B peptides. As a result, three amino acids, that is histidine, asparagine, and serine, that are far apart in the primary sequence are aligned at the active site and take part in the catalytic process. All the gastrointestinal enzymes are synthesized in the form of proenzymes, and only after secretion into the alimentary canal they are activated. This prevents autolysis of cellular structural proteins. Coagulation factors are seen in blood as zymogen form. Similar activation of precursor protein is taking place in the case of complement components. These activities are needed only occasionally, but when needed, a large number of molecules are to be produced instantaneously. Hence, the cascade system of chemical amplification of such factors is seen. The next factor is enzyme inhibition. In this, competitive inhibition will be first. Here, inhibitor molecules are competing with the normal substrate molecules for binding to the active site of the enzyme because the inhibitor is a structural analog of the substrate. E plus S bidirectionally gives ES complex, which finally gives E plus P, that is enzyme plus product. On the other hand, E plus I bidirectionally gives EI complex, and the reaction is stopped. Since the EI enzyme inhibitor complex can react only to form the enzyme and inhibitor, the number of the molecules available for ES formation is reduced. Suppose 100 molecules of substrate and 100 molecules of inhibitor are competing for 100 molecules of enzyme. So half of the enzyme molecules are trapped by the inhibitor, and only half the molecules are available for catalysis to form the product. Since effective concentration of enzymes is reduced, the reaction velocity is decreased. In cooperative inhibition, the inhibitor will be a structural analog of the substrate. There will be similarity in three-dimensional structure between substrate and inhibitor. For example, the succinate dehydrogenase reaction is inhibited by malonate. In this diagram, we can see that succinate is converted to fumarate, while malonate is a structural analog of the succinate, which inhibits the reaction. Competitive inhibition is usually reversible, or excess substrate abolishes the inhibition. In the previous example of 100 molecules of E and 100 molecules of I or inhibitor, if 900 molecules of substrate are added, only 1/10 of the enzyme molecules are attached to the inhibitor and 90% are working with substrate. Thus, 50% is the first example is now decreased to 10% inhibition. In this diagram, you can see that 1 upon concentration is plotted against 1 upon velocity, where without inhibitor and with inhibitor, the substrate constant saturation is present. In this graph, we can see that the substrate concentration is plotted against Vmax, where with inhibitor and without inhibitor, the plot is obtained. From the graphs, it is obvious that in the case of competitive inhibition, the Km is increased in the presence of competitive inhibitor. This competitive inhibitor apparently increases the Km. In other words, the affinity for the enzyme towards the substrate is apparently decreased in the presence of inhibitor. Here ends our session. Thank you. Please like and share and subscribe the channel.